This approach allows for the quantitation of known PFAS and the discovery of new substances in the same samples, compared to targeted approaches alone that miss emerging compounds of concern. This protocol targets a wider variety of PFAS compounds, which are representative of the newest emerging chemicals. If the concentration step is adapted for the compounds, the measurement methods used here could be applied to any class of chemicals.
To collect a sample from the field, wear nitrile gloves and collect 500 to 1,000 milliliters of water from the field location in a clean high-density polyethylene HDPE or polypropylene bottle. Add 5 milliliters of 35% nitric acid preservative to the sample bottle and to a field blank, and transport the samples back to the lab. Pour each sample into individual pre-cleaned 1-liter high-density polypropylene graduated cylinders and record the exact volume of the samples. Add 10 milliliters of methanol to each emptied sample bottle before capping and shaking vigorously to rinse absorbed per- and polyfluoroalkyl substances, or PFAS, from the bottle interiors. Then, return the measured water sample to the rinsed bottle with the methanolic rinse. To obtain a standard curve for quantitation, fill 8 empty 1-liter sample bottles with PFAS-free deionized water and label the bottles with 8 evenly spaced concentrations covering the desired quantitation range. Next, add a quantity of native PFAS mix to each bottle to yield the appropriate PFAS concentrations, and add the internal standard PFAS mixture to each sample at a concentration approximating the midpoint of the calibration curve.